All right, another great presentation. We are going to keep it rolling, get back to the schedule, and try and stay on time. So we're going to bring on our next speaker of the day, John Kovach, MD, CEO, and founder of Lixte Biotechnology Holdings. John, how are you doing?
Very well. Thanks, Michael.
Thank you. Good to have you here, John. I know you've got a presentation ready that we're going to be pulling up with slides, so we'll get that on, and the floor is yours. Take it away.
Thank you very much. It's a pleasure to have an opportunity to tell you a little overview of Lixte Biotechnology and why we think we have something very special. Today, I'm just going to give you a quick overview of where we are with our clinical lead compound, LB100, and then spend a little time discussing why our recently announced collaboration with Netherlands Cancer Institute based in Amsterdam we think is so exciting and potentially very valuable to us as a company, as well as to future cancer patients. Next slide, please. I think it's just, of course, I'll have some forward-looking statements in this presentation. And next, please. Just in brief, for an overview, we've designed and patented a series of novel drugs that enhance the effectiveness of standard anti-cancer therapies in a variety of animal models. There are over 40, closer to 50 now reports that our lead compound, LB100, increases the anti-tumor activity of standard chemotherapy, radiotherapy, and the exciting new avenue, immunotherapy, without increasing toxicity. An incredible finding, and one which has been borne out by the repeated studies, as I've mentioned. We're now at a clinical stage of showing that what's holding true in the animal models holds true in human studies. But next, please. Interestingly, we're not aware of, at the present time, any competition to our lead compound. Why we think people will be after this soon, fact is that because of the sweeping scope of our compound, it's really been very difficult to convince people that we can have something that increases the three pillars of cancer therapy, that is chemotherapy, radiotherapy, and immunotherapy, because the market would be extremely large, to say the least. Next, please. How is it possible? How can LB100 work and have all these effects without increasing toxicity? At a simple level, one can think of all cell activities require switches to rapidly turn on and off biochemical pathways. This is a matter of seconds to minutes. In all cancer cells, a major driver of the cancer process itself are one or more, generally many, defects in these switches. Next, please. Our compound, LB100, inhibits one of the master off switches, which is an enzyme called protein phosphatase 2A that regulates many aspects of cell growth, repair of DNA damage, and regulation of T-cell immune function. If you just think about it for a moment, I mean, those are the three main targets of virtually all cancer therapy at the present time. How to alter cell cycle, that is cell growth of the cancer cell, how to interfere with the ability of the cancer cell to repair its DNA damage induced by either chemotherapy or radiation, and how to unleash the human immune system from the restraints it has on T-cell function. T-cells are those that recognize foreign invaders and kill them, and as a basis for the remarkable advances in immunotherapy. How can one compound affect all these activities? Next, please. In fact, there's considerable skepticism. We ourselves entered a phase 1 trial giving LB100 as a single agent, even though we know it's most likely to be most effective when combined with other drugs. And to our pleasure and excitement, we found that one can administer LB100 at doses which are readily tolerable, not causing essentially any toxicity, and yet be associated with effects on cancer cell growth. During that trial conducted at Mayo Clinic, City of Hope, in the U.S. Oncology Network. One patient with advanced pancreatic cancer who had failed now 4 or 5 separate individual treatments had an objective response lasting almost a year, receiving a relatively low dose of single agent LB100. Mechanism is still not exactly clear of why that's the case, and yet 10 of 20 other patients in that trial who had measurable disease had cessation of growth of their cancers for several months. Really, it doesn't sound like much, but several months in patients with advanced progressive disease is quite remarkable for a single agent that's causing no toxicity whatsoever. So that was the real breakthrough. That's what gave us the encouragement to push on and go into clinical trials. Next, please. Now here's our pipeline. That is what we've really been doing at the clinical level. I'm going to really emphasize first 2 clinical trials, one in lung cancer and the other in sarcomas, but to touch base on a couple of other studies which are ongoing, just to bring you up to date for those of you that have been following Lixte. They're moving slowly, but are very, very interesting. They're really a little bit off the mainstream of where we are with our current trials. First is myelodysplastic syndrome. Myelodysplastic syndrome is a disease of the bone marrow in which generally elderly people, bone marrow starts failing, and the problem is developing severe anemia requiring 2 or 3 blood transfusions a month to maintain their hemoglobin at a reasonable level. We were approached by Dr. Alan List, who's one of the leaders in this field, then at that time the head of the cancer center at Moffitt in Tampa, who wanted to study our compound and did preliminary studies in the laboratory showing that in preclinical work, LB100 as a single agent was more effective than Revlimid, the lead compound, the only one approved for a subset of myelodysplastic syndrome. This was quite extraordinary, and we signed an agreement with Moffitt to allow them to proceed with a single agent trial in myelodysplastic syndrome using LB100. The agreement includes a small royalty payment if it's successful. This trial is moving extremely slowly because the patients are quite well. And during COVID, the demands of coming often to the hospital to receive a single injection was somewhat limiting. But it's proceeding. It's on the side. It's costing us very little to continue that trial. And if indeed, as the preclinical studies show, LB100 is active, it will be a significant advance in a relatively rare but important disease. Also, similarly, and not really in a therapeutic trial, but we were approached by the National Cancer Institute, as at NIH, to see if we would join with them in doing a pharmacology study of LB100 in brain tumors. Brain tumors of adults, the lead one called glioblastoma multiforme is one of the most aggressive and uniformly fatal diseases of human beings. They wanted to see whether LB100 entered the brain in sufficient quantities to be active. The reason this is important, the reason NCI is interested, as are we, is that LB100 has been shown repeatedly to potentiate radiation and the drug temozolomide. Radiation and temozolomide are the standard treatments for initially treating glioblastoma multiforme. And if LB100 could enter the brain in sufficient quantities to potentiate them, we hope this would be a significant advance in this disease. That study is complicated. It could only be done, really, I think, very few places in the world. One of them is at the center in Bethesda at NCI, where patients with recurrent gliomas who would be eligible to have recurrent disease resected are taken to the operating room for resection of 1 or more recurrences in the brain. In the study design, those patients on the day of their surgery receiving 1/3 of a full dose of LB100, as we know it's safe. And then the behavior of the drug, called the pharmacokinetic behavior of the drug in the blood, and in the tissue that's removed can be observed. That trial, again, has essentially been stopped because of the COVID epidemic necessitating closure of the surgical unit at National Cancer Institute, neurosurgical unit. We understand that that is recently opened and accrual will resume in gliomas. So we're optimistic. 2 patients had been entered, as we've reported in our 10Qs. We're optimistic that we will achieve the 8 patients goal before analyzing the results in the relatively near future. But I'm going to spend a little more time today talking about small cell lung cancer and our plans in soft tissue sarcoma. Next slide, please. We see small cell lung cancer as an ideal target for efficiently testing the ability of LB100 to improve the efficacy of standard chemotherapy against a very aggressive human cancer. Next, please. Small cell lung cancer is one of the most aggressive of all the lung cancers. If you think about it, with the best therapy, treating previously untreated patients with the best therapy, time to progression of disease is only just a little over 5 months, and median overall survival. People live only for a year, and the median, just about a year. In all animal models of small cell lung cancer, it's been demonstrated by ourselves and in working in conjunction with others that LB100 is active by itself and increases the effectiveness of the standard regimen. It's interesting that it's active by itself in the small cell lung cancer. All small cell lung cancers have a particular genetic defect, which renders them particularly susceptible to inhibition of PP2A, the target of our compound. This has recently been published by a group including ourselves and investigators at the City of Hope National Medical Center in Duarte, California. Next, please. We were approached by Dr. Ravi Salgia, Chief of Medical Oncology and Lung Cancer Expert at City of Hope, who designed a phase 1b trial to see whether LB100 improves standard therapy of small cell lung cancer. I'm spending time on a phase 1 trial because you usually say, well, gee, that's so early, it's only usually safety trials. But in this particular trial, previously untreated patients in groups of 3 received standard therapy, full dose standard therapy, and then escalating doses of LB100 to determine just how much LB100 can be added and see if it's really potentiating standard therapy. When the maximum dose of LB100 is determined, essentially is a phase 2 component in which additional patients, excuse me, in this case, 12 additional patients will be treated at the phase 2 dose. If indeed LB100 potentiates the elements of the standard therapy in which it comprised this regimen, we fully expect to see significant activity in those 12 patients, enough to merit extension into phase 3 trial, and even in our fantasy, if it improves therapy by 50% or more, to extract excitement as to proceeding immediately to a more definitive clinical trial, if not actually to approval to sell the drug for that purpose. It's a big reach of a small trial, but we're very optimistic that we expect to be able to see in this group of patients where LB100 truly lives up to all the findings in preclinical trials potentiating cytotoxic chemotherapy. Next, please. The other group tumor we're focusing on, soft tissue sarcoma. Soft tissue sarcomas are equally highly aggressive cancers for which there is really no effective treatment. We're limiting this study to 2 sarcoma subtypes which progress on the average, this is from initial treatment, of less than 5 months of beginning initial therapy. A single drug, chemotherapy agent, doxorubicin, has been the standard treatment for over 40 years. In several animal models of soft tissue sarcoma, LB100 again markedly enhances the effectiveness of doxorubicin. Next, please. And we were approached by Spanishsarcoma Group has been for about 20 years trying to find new approaches to treating this disease, whether we would work with them in doing, in this case, a randomized trial in which patients with these very aggressive forms of soft tissue sarcoma receive standard therapy, doxorubicin, with or without LB100. The goal here would be to see if the significance would be whether there's increased progression-free survival as the disease was kept in check from an average of 4.5 months to 7.5 months. A very modest demand, we think, and one we hope we'll be able to see in this randomized study. Now, we've had even though the trial has been approved by Spanish authorities and by all the centers in Spain which are interested, including some other centers outside of Spain which would like to join this trial, by the insistence of EU regulators that we've synthesized a new batch of LB100, which meet the current EU regulations, for drug synthesis. Batch we're using in the United States is just fine, but the European authorities are required we make a new batch which has been made and which is currently undergoing quality control in Europe. We hope that we'll be able to open that randomized trial sometime in the earlier to mid part of next year. Next slide, please. But what I want to spend a little more time on today is the recent collaboration we've announced with the Netherlands Cancer Institute based in Amsterdam. Awfully sorry. We were approached by Professor René Bernards, who is a world leader in molecular and genetic techniques in identifying effective drug combinations, to see if we'd be willing to collaborate with the Netherlands Cancer Institute around LB100. Professor Bernards has used various genetic screens in the past to identify, in this case, one now FDA-approved combination of treatments for a particular type of colon cancer in which there's a certain mutation. Unexpectedly, he was able to find, using his screening techniques, which subset of patients with colon cancer would be vulnerable to this particular drug combination, leading to its approval for therapy. It's now being approved, now being used throughout the world. Bernards would like to apply his unbiased genetic approach to identifying the best combination of LB100. It would be a great help. As we know, we picked two, sarcoma and small cell lung cancer, which clearly in model systems should be very sensitive to LB100 addition. But there are many compounds likely to be that we could never pick out just on the basis of their known activity as to whether they're going to be useful with LB100 or not. Next, please. So the advantage of this collaboration gives us the opportunity, working with Professor Bernards, to identify unsuspectedly effective therapies that use LB100 to potentiate standard chemotherapy. That is, of standard agents which are already approved and being used. So the time to application would be quite short. It also gives us the opportunity to identify new uses, potential new uses for investigational drugs, which have been studied extensively but found to have only marginal therapeutic effectiveness on their own. If this were the case, this would markedly expand our opportunity for new patents on combinations of LB100 and create an opportunity to enlarge our pipeline, extending well into the future. A very unusual opportunity, and one which we're excited to begin with Professor Bernards in Amsterdam. Next, please. Thus, it's a quick overview. It's a lot to digest in a short period of time. Our lead compound increases the anti-tumor activity of chemotherapy, radiotherapy, and immunotherapy without increasing toxicity. A remarkable situation, one which we must prove applies to human beings in the planned and ongoing clinical trials. But if the preclinical results hold true with just one cancer in the clinic, feel no doubt that LB100 would be truly a game changer in cancer therapy. Thank you very much for your attention. I appreciate the opportunity to present today.
Fantastic. John, thank you. We always appreciate having Lixte with us, and that was an excellent presentation. One thing maybe before we leave and move on to our next presentation, if there's one thing you could leave investors with, the Benzinga audience as well, that you're looking forward to on the horizon, give us just that one piece that you're most excited about Q4 leading into 2022 as well.
Well, the results, we are looking to add additional sites to our small cell lung cancer trial to make sure that accrual is robust over the next year and a half, because as I pointed out, with a very small number of patients fully treated with our combination at the full dose, trace 2 dose, we really anticipate to be able to prove that what we found in preclinical models applies to human condition in a terrible disease in which evaluation can be done in a short period of time. Thanks for that question. That's where we're headed. Of course, we don't know what's going to be revealed by studies in Holland, but they're underway, and we hope to have a lot more exciting things to report in future conferences. Thanks, Michael.
Thank you, John. We appreciate it. A lot to keep an eye on. We will definitely have it on our radar. Looking forward to talking with you again soon. You take care.